Hello everyone. Today we are going to discuss the topic antibody. Antibodies are immunoglobulins. It's a Y-shaped structure. It is made up of glycoproteins. It's also called as B cells that specifically recognize antigens. The main function of the antibodies is neutralization, obscuration, and complement activations. They have a characteristics of four chain structures with defined primary and secondary structures like IgG, IgM, IgA, IgE, and Ig. The chemical nature of glycoproteins are it's made up of glycoproteins. The molecular weight is 150 kilodalton in having two heavy chains and two light chains. The heavy chains will be containing 50 kilodaltons and light chains will be containing 25 kilodaltons. These two chains will be connected with the help of disulfide bonds. The specificity of the antibodies are bind to a particular antigen with a high affinity, minimal cross-reactivity, unique amino acid sequences within the variable regions. The affinity of the antibody is strength, which is interaction of antigen and antibodies. Higher affinity antibodies binds more tightly effective at neutralizing or eliminating antigens. Avidity means overall strength of interaction of antigen and antibody. For example, IgM with its 10 binding sites exhibits high affinity even if the affinity of each individual binding site is relatively very low. The biological function of the antibodies: neutralization of toxins and viruses, opsonization for phagocytic cells, especially IgG antibodies. The complement activations, mainly IgM and some IgG subclasses, are mainly involved in the complement activation process and also involve the antibody dependent cell mediated cytotoxic cells, which will be used to neutralize the cell infected with the antigens, which recruit natural killer cells, release cytotoxic animals that kill the infected cells. The primary structure of the antibodies, which will be contain the amino acid sequences, specific linear order of amino acids in heavy chain and light chains determine the antibody's unique properties. Key region, variable region, and constant region are there, which contain one variable domain and typically three or four constant domains. Light chain, one variable domain and one constant chains. Hinge region is a proline reach region in heavy chains that provides flexibility separating the antigen binding and effector parts. This is the complete structure of the antibody having the two chains, that is light chains and heavy chains. It containing the one variable region and three constant region, and light chain contain one variable region and one constant region. This is the hinge region which connecting the two heavy chains. The second structure of the antibodies is containing the backbone is containing local folding of the polypeptides. Backbone is the repeating pattern through hydrogen bonds, which contain the beta sheets, the dominant secondary structure forming stable platform for the variable and constant domains. Beta turns and loops connect the beta strands, especially important in variable region for forming the antigen binding loops. These are the two scientists, Gerald M. Edelman and Rodney R. Porter. They were got the Nobel Prize in the year of 1972 for the identification of antibodies. Thank you.